Please allow me to start recording with permission of Gaurav, and
Yeah.
please allow me to introduce Gaurav. So in Sciversation with Prash has been a monthly conversation with scientists who have traversed toughest paths. In fact, this Sciversation I usually do it with those particular people whom I knew over the last at least 3 years or more, and this is typically a kind of an interwoven story in such a way that we ask certain unasked questions, absolutely pretty extempore, pretty open, and we usually invite the speaker to present for the first 10 minutes. So today Gaurav
Mhm.
is going to present for the first 10 minutes about his life journey. Gaurav is a PhD from JNU way back in 2016. Previously, he has done his MSc, and then he went to UCD, University of California, Davis, in the States, where he did his excellent postdoctoral years of experience. And he returned back in 2019, I believe, as a
Yeah.
DST Inspired Fellow with his own fellowship and a major grant, and then he joined IBAB Bangalore, the prestigious IBAB Bangalore. That's where I started knowing him in 2019 because मैं हमेशा जब भी मैं सोशल मीडिया देखता हूँ तो यूजुअली मैराथनर्स को देखता हूँ, and Gaurav was came into my wonderful eye capture. He was an absolute, he's still an absolute marathoner. He does his full marathon 41.2 kilometers, not just in India, across different terrains and across different things, but also he went to Paris recently. He did that kind of coverage over there. और मेरे जैसे बंदे बहुत jealous हो गया कि how come I mean मुझे लगा कि he might have run all the way from maybe Hyderabad to Paris, और मुझे लगा कि फ्लाइट में भी वो जॉग करता रहा, that was how I felt. But he's such an amazing person. I met him 3 or 4 times, and very recently was in Hyderabad itself, where he has taken his assistant professorship, and his group works on microbial genomics, host pathogenesis, and intermittently works on the plant microbial things. They have developed some wonderful, models, both genomic and bioinformatics models, taking up, the precision scale, for this microbial genomics, which he'll probably roughly discuss. And most importantly, he has developed a wonderful Google Scholar-like suite, which he calls this as SG Index, which is going to present it today, during this Sciversation. Absolutely honored to have you, Gaurav. Thank you so much. So please share your screen and make your presentation. Thank you so much.
Okay, thank you very much, Prash. Means it's really an honor. Means when you messaged me about this, the first thought was that okay, definitely I'm not eligible to give such an interview because you usually take very prominent researchers. So, but yeah, still honored, and means it's a great initiative, and I'm sure people here will learn a little bit of something new. I hope. I hope you can see my screen now.
Yeah, yes, please make it full screen, Gaurav, please.
Okay, so yes, this is visible, right? Uh-huh. Okay, so this is my lab, a small lab with several PhD students and several interns. And I usually call my lab more of a knock-knock lab. Knock-knock lab because we try to understand signal transduction, and we work with microbes. So we usually go to each and every microbe and try to ask, who's there, what they are sensing, and how they are sensing the environment, what are the tools and techniques they have. That's why I call it like usually knock-knock lab. I think some of the speakers are on. Huh. Okay. So what we usually do, we study life via trees. And I hope many of you remember this figure. This is from Darwin's book. And he was the first guy who introduced these kind of phylogenetic trees and how they can be used to understand life. So, but now obviously there are so many different tools, so many molecular markers which we have been using from last 20, 30 years, and we make these kind of trees for using those markers and try to understand the diversity and the different type of functions altogether. Prash has already mentioned about my journey, but just to give you a brief introduction, that I did my master's in biotechnology from Jawaharlal Nehru University School of Biotechnology. I worked with Dr. Swati Tiwari there, and initially my work was on experimental work, where I was planning to do some northern blot to understand gene expression in anti-histolytica, and it was a very big failure. Means I was not able to perform anything there as such. So in the last 2 3 months we decided that, okay, let's try to do some bioinformatics work, and everything start falling in line, and we had some good data in those 2 3 months, and then later on I worked for 2 3 months after the master's course, and we even published that work. So that was the initiation point for me in bioinformatics field. So that's why I say that I'm not a bioinformatics by training in a way that I did my bachelor's and master's in bioinformatics, but I started doing bioinformatics in my PhD. And in my PhD, also my target was to join a lab which is prominently doing bioinformatics or in silico work. And I joined Dr. Sri Krishna Subramanian's lab at IMTech Chandigarh. He was a protein bioinformatics, structure bioinformatics person, but we started doing a lot of microbial genomics during my PhD. And then after my PhD in microbial genomics and evolution, I went to UC Davis for my postdoc. And there also I worked with genomics, transcriptomics, and evolution kind of studies. Came back to India after 3 years in Bangalore as a DST Inspire faculty. And we did some good work during my DST Inspire faculty time period at IBAB. And then I joined Department of Biotechnology as an assistant professor like 3 years back. So this is kind of a quick journey about me. And now we have Sharma Genomics Lab, where there are many people who are working on several very interesting questions, which I will discuss a little bit. So in case you haven't visited Hyderabad, so this is IIT Hyderabad is kind of located a little bit outskirts of Hyderabad. And this is our biotechnology and biomedical engineering building. It looks a little bit rusty, but that's how the building architecture is. And there are multiple divisions in our department, and I prominently work on big data biology and microbiology, and we try to do a lot of things in both. If you want to understand what we usually do, we work a lot on sequence, structure, function, and evolution. And this is like the broad four terms, and most of our work lies in one or multiple of these directions. And a very fascinating field to work on, where there is a lot of open access data, and obviously you can make your own data using sequencing. So a lot of scope in this particular field, especially for the young people who are here, including me, obviously. And these four words are actually the holy trinity kind of, or tetranity, you can say, because sequence springs to structure, structure emerges function, and function flows into the evolution. So these four words are very broad and covers like all the dimensions of biology, frankly. So some focus areas of my lab is we work a lot on microbial genomics and evolution. I will not discuss like my science as such. Obviously, you guys can go through it and find out some very interesting things which we have been doing. But just want to give you like broad ideas what we have been doing. We are also doing medicinal plant genomics and metagenomics, where we are trying to understand plant microbe interaction using plant genomics and metagenomics. We do a lot of collaborative work. And we are also developing several fascinating tools, which are ideally very basic idea based. We are not trying to do a lot of high data analysis kind of tools, but more of where we see some lacuna, we try to develop those things, which are part of the PhD project of those students. So, yeah. These are some of the model organisms which we have been working on. And as I mentioned that myxobacteria is one of the prominent model organisms for which we have been working. I have been working from last almost 10-12 years since my PhD. And in between, we also worked on SARS-CoV-2. We are working on medicinal plant, Vibrio, and obviously metagenome or microbial communities. Some of the broad ideas which we have been working on are especially in the sensing and motility evolution, that how let's say myxobacteria have evolved sensing mechanisms and how their motility is evolving. We are also working on polysaccharide biosynthesis pathways, secondary metabolites, and so many different things at a specific bacteria level, let's say myxo, SARS-CoV-2, Vibrio, and some, and also about like when we study all microbes all together and try to understand their diversity. So as I mentioned that we work on myxobacteria, they are very fascinating organism. If you see their genome size, because that's what matters to us, because we work with genome, they are the highest genome size organism within the bacterial kingdom. And that is one prominent question for us, that how they gained this much of genome size overall. And obviously, along with this genome size expansion, they have developed so many fascinating physiological characters like fruiting body, chemosensory system, gliding, wolf pack hunting, so many. And they are non-pathogenic. So that is the most interesting aspects about them. And we are trying to understand all of these things in a diverse manner, starting from genome sequencing, strain reclassification, signal transduction, gliding motility, development cycles. We are either in collaboration or in independent efforts. We have been working on several of these dimensions to understand that how myxobacterial physiology and evolution has been going on. And still many students are working on this front. During when I was a PhD student at IBM, we also, and similar to everyone, we are professional biologists. We were working on coronavirus also, and we did several studies in terms of understanding their diversity, understanding their evolution, and using whole genome and genome perspective. Did some policy research where we said to de-escalate all the countries that there is a huge unmet in huge and countries should try to sequence these organisms quicker so that the variant can be identified immediately. At that time, when we published this work, India was doing that at like 7 days on median scale. So it's like almost 2 months time they were taking from strain collection to submission. And in contrast, USA was taking like 7 days or 10 days. It was a huge gap. We tried to report it, and this was at that time prominently published and cited so many wonderful researchers across. So the tools we have been taking since last 1 year only, or actually 1 or 2 years, not more than that, and they are very fascinating to some of them, like the case scholars try to discuss media. And recently, this work is under intensive.
Yeah, Gaurav has left. I think he has a connection. He's somewhere in Bangalore, so um, he'll be back. Huh? Yeah, I think he must be joining back. Yeah, voice is breaking with him. It's also for me. Um,
Yeah, yeah, no problem.
Actually, he's in retreat. In Bangalore in
Yes, yes, yes.
Aurobindo's ashram. So he was.
Mm-hmm.
So
No, once in a while that happens.
yeah,
Yeah, that's okay.
yeah. Welcome back, Gaurav. Yeah, yeah. So on a lighter note, signal transduction event has happened. That's the reason why you left.
Signal transcription in self board signal.
Yeah, in safe mode signals and do. Yeah, that's a great point. Yeah. Gaurav? Yeah.
I'm here.
Yeah, yeah, please. Yeah. Thank
Okay.
you. Yeah.
Yeah. You can hear me, right?
Yeah, yeah, please.
Yes. Prash, I think you are on mute.
Yeah, I'm muted. Yeah.
Okay.
Oh, हो गया क्या आपका presentation?
Yes, yes, yes.
Oh, I'm sorry. I'm sorry. I thought it
Yes.
was slides. Okay.
Uh-huh.
Yeah. So thank you so much, Gaurav, for a wonderful presentation. I think that was a great kickstart. I mean, I was always fascinated about your wonderful paper on SARS-CoV-2 with the lag data sets that were published in Lee of SARS-CoV-2. So, just please tell us the background about that particular work, Gaurav. What was the origin of that particular work? That was a wonderful work. Let me be very open. I read it at least twice. Yeah.
Right. No, thank you, thank you. So, means ideally, that's a very simple analysis. Means I must say, okay, because and it was only about the idea that it went somewhere like Nature Biotech, because ideally, how it started, there was a lot of when SARS-CoV was happening, there was a lot of rumor about not rumor, but the real incidences about different kind of variants, and I was just, wondering and confused at one point of time that, okay, how much time we are taking in identification of such kind of variants. Okay, so I did a very normal study in the night when everyone was sleeping. I just woke up and tried to just did some comparative between India and UK, and I found out, okay, the median is like so much varying between India and Japan, India and UK. For UK we were getting like 10 days or 7-8 days, and for India it was like 61 or 70 days. So I woke up next day. I talked to my wife about it. That okay, means my wife is also a researcher and scientist. So I talked to her. That then she said that okay, so what is new about it? Everyone knows about it that India is a developing country, and obviously we will take more time, and obviously UK and USA will do it like quicker. I said that okay, means everyone knows about it, but no one has reported the number as such. That how much every country is doing. So then then I thought that okay, let's try to do this wholesome analysis based on GSID data, and we did it in like 7-8 days. One of my student Kishan helped me out in that, and ideally it was like 7-8 days work, and then we started talking about the results. I talked to another colleague, Gayatri Sabharwal, who was doing a lot of policy type of work, and we tried to reframe the whole idea into an article. And yeah, submitted there, obviously waited for 5-10 days. They didn't message and anything. And then they said that, yeah, it looks interesting. Why don't you do some kind of modification and then resubmitted it? So it was a very good experience in that way, because the ideology wise, we have simply done one calculation. That's it, subtraction, and nothing more in that.
Right.
Because it's just that when the sample was collected and when it was submitted to JSID, we have just subtracted these two numbers and then calculated the median.
Right. And
So yeah.
I still remember, Kishan Kalia was telling me, because he was my student at that point of time, he had a wow moment, after going, to your lab in IBAB. He said कि sir, इतना सीखने का मौका मिलता है मुझे, गौरव से, and I really learned a lot. And at that point of time, आप यकीन नहीं करते हो गौरव, you are probably trying to be largely modest. आपका publication देख के Indian submissions of COVID बहुत बढ़ गया, actually.
Yeah. Means, it impacted. Means
Yes.
there were discussions in like in big levels also
Right.
that what is happening and all.
Largely with the government sectors. Yeah. Thank you so much. Yeah. And the reason why I made that particular point was we also sequenced about 900 SARS-CoV-2 genomes. With our collaborators, we have done metatranscriptomics, metagenomics. We also had a lag phase at that point of time, and that was very fascinating. That lag was not just because of lack of what do you call data sets or time, but because of logistics reasons. Because SARS-CoV-2 consortium, they were not decided what to do with those data sets actually.
Yeah.
So that was
That's
also one of those reasons. Yeah. So second question was, Gaurav, you have done almost more than 15-20 genome sequencing projects. That was quite wonderful. If you really see across different things, that was quite wonderful. One thing that always fascinated me was you have done, of course, the whole genome sequencing, assembly of these microbial samples. You also put up a very amazing effort on composite genome sequencing, as opposed to something what we call it as a pan genome sequencing. So what is the rationale behind a composite, the word composite in this particular case, as opposed to a pan genome, at this point of time?
Yeah. So I think so traditionally, like people do single strain sequencing, and we have also done a lot of single strain kind of sequencing. But what happens, like in some cases, it's very difficult to, separate multiple strains which are very similar to each other. So let's say in terms of, we did one sequencing project on Bacillus clozy. So this Bacillus clozy was, means I'm sure all of us had at one point of time taken this Enterogermina probiotic. Okay, so Enterogermina probiotic is a collection of four Bacillus clozy strains which have different antibiotic resistance. So it was a very tough job. Even now, people have not been able to separate them from Enterogermina directly. So when we submitted it, we thought that okay, it's not one strain sequencing; it is four strains which we are submitting. So that's why we say it a composite strain, composite genome sequencing for that particular Enterogermina. But in terms of pangenome, what people, it's even pangenome can be done in the similar manner. But in that situation, usually the organisms can be different at species level also, at genus level also, at different levels, right? So in that way, that is a little bit different. What will be the main point here, that if the strains are species level different, then to culture them it easy, because they will have some different type of morphology on the plate. But if they are different at strain level, and at strain level very minute difference, that in one of the organism in those four, the difference was only at one nucleotide level in RPOB gene, only one nucleotide level in RPOB gene, and that was the only difference. So in that way, it will be very complicated to separate them. But in pangenome case, it's possible that you can culture them and differentiate them separately also.
Wonderful.
Yeah.
What a great point, Gaurav. Yeah. And seldom do we find scientists or researchers working on evolutionary pathways after sequence structure functional relationship. And probably your lab is pretty unique in looking or in going an extent forward in looking into the evolutionary landscape after the functional entities are done. So I was just wondering, there is of course a big cross link between signal transduction with respect to the microbial crosstalk, and also at the evolutionary scale there could be lateral gene transfer events as well that might be afflicted with probably signal transduction as well. So did you find over the years any kind of clues where you might expect that TK, there is a lateral gene transfer or horizontal gene transfer event that is afflicting the state of the art, what is happening with signal transduction events. So are there any clues?
Yes.
In that?
Yes. So we have been working on signal transduction in Vibrio and in Myxobacteria, like very largely, and we are seeing those kind of events happening. Because see what happens when we have such a large data set. So let's say for Myxobacteria, we have one unpublished work which we are planning to submit very soon. So we have worked on like almost 100 pure culture genomes and almost 3,000 metagenomes. Okay, and what we have found that that there are one or two specific signal transduction full cluster, like full chemosensory cluster, which has been acquired by some of the organism, and they inculcated it into their genome and into their expression, and have been using it very wonderfully. Right. So let's say when we talk about Myxobacteria, or let's say when we talk about E. coli kind of organism, E. coli is we usually say it's a very, I will not use the word basic organism, but I will say a highly evolved and highly studied organism. And it has only one chemosensory system, which helps in motility. Okay. Now Myxo has 8 to 12 chemosensory systems just to govern different type of motility, different type of fruiting body formation, might be flagellar motility, or so many, like even governing biofilm formation or sporulation. It might be doing so many different functions. So they have procured them from somewhere, and later on, after the procurement, they have incorporated them into their life cycle in such a way that the regulation of the mechanisms or the physiology becomes easier. Okay, so similarly in Vibrio, we have found one more extra chemosensor system which people have not reported till now. We are again trying to publish that work very soon. So, and the important thing is that such kind of evolutionary event doesn't happen all across the organisms. They happen in a pocket of organism. Okay, so let's say in Vibrio, if there is a fourth type of chemosensor system which is present only in few organisms, not in all the organisms. So it means that for some organism where the necessity was, they kept it, and for others where the necessity was not there, they didn't kept it and they just let it go. Yeah.
Wonderful, wonderful. Yeah, what a great explanation. Thank you so much, Gaurav, on this. One minute, sorry. I don't know. Um, yeah. So moving forward, Gaurav, that begged us a question, Gaurav. Last week, in fact, we had torrential rains here. A large number of rains. और मेरे फ्रेंड्स बहुत लोग कंप्लेन कर रहे थे कि मैं केरला से बहुत सारे रेन्स लेके आया जयपुर. And then I started wondering, the roads were so bumpy, आप यकीन नहीं करते हो कि इतना गड्ढे हैं, and there were other things. And we were discussing on several fronts, what could be a likely possibility to ensure that the roads can be bettered. तब मैं कहीं पे मैं पढ़ रहा था कि there are of course lactases which are well known enzymes that are microbial things. I think Vinod sir is there, he's an expert in that. In fact, I thought of discussing with him as well. Or these lactases can be enhanced by producing them from good microbes, where they can be converted into those paper paste, and these paper paste can be again fabricated in the form of a plastic, and these particular plastic can be made in the form of a roads.
Yeah. People have been thinking.
So then that begged us a question कि जब आप ये इतना बढ़िया आप explain कर रहे थे अभी, so there could be some ways to ensure that we find those particular cascades that can ensure that such kind of things can be taken up, and of course that can really do that. I don't know why it's a very simple thing. That's a wonderful, probably a good project to think of as well. That was what
I think there are a few limitations to this, because those organisms which we are using for such kind of thing, they should also not be toxic or not involved in some other activities, and that might be hindering their progress and something like this, right? So because see, like an organism doesn't just make one property, and one property can be complemented or like in the new in a reverse manner, complementing it to some other purpose, right? So we need to see all the things together or at a meta scale to know that, okay, if we are planning to use this particular organism for this activity, it may not be harming us in some or the other manner.
And chemical sensors induced by them would also be different,
Yes.
Yeah. So that could also be, that's a great.
Sometimes it has also been seen that one organism behave differently as alone and differently in a swarm or in a group of organism, right? So that is also one other thought which is worth considering.
Yeah, wonderful, Gaurav. Moving further, what was the question? If you really see, you also worked on the prokaryotic nomenclature system. In fact, I think I faintly remember I discussed this with you last April. This April. Because I also work on hypothetical proteins, and we recently started over the last 6 years. We dealt on the virulence factors, hypothetical protein classification methods. So there could be a stringent nomenclature system, for those chemical sensor induced genes. जैसे GenBank में update हो जाता है तो there is no a coherent nomenclature system that honors
for organism or for organism you are saying?
I'm talking for the enzymes. Sorry, for the enzymes.
Okay, for enzymes. Yeah.
So although EC derived classification is quite good, but if it were a protein encoded by a gene, we really don't have a stringent nomenclature system as we have for the human genome, human genome organization. So, so can you please tell us something about that wonderful paper of yours, Gaurav, on the prokaryotic nomenclature system that you have worked on?
Yeah, yeah. So basically, what happened at that time? So many of my ideas or many of my such ideas are highly influenced from social media. Okay, means I give like a lot of credit to social media because at one point of time, Twitter was very informative and there were a lot of discussions on so many different things. So what happened in 2021? 20 or 21. So this prokaryotic classification team, they changed the whole prokaryotic phyla names. Okay, like let's say um,
Yes,
bacillus.
subspecies classification we change kar diye. Yeah, you're right.
Yeah, yeah.
Yeah. Okay.
Means, in a long run, obviously that was a welcome step, frankly speaking. But it also changed. They changed like some things very drastically. Okay, like let's say proteobacteria. They could have made it like proteobacteriota or something like that, right? But they changed it to let's say I think pseudomonodota, okay, just based on one organism. So there were a lot of hush about it that, okay, why they are doing it. And so we thought of writing something and so I'm sure like we were also a little bit delayed on that front. Initially, I was not planning to write about it. And later on, we thought like when someone published something in that direction, we thought of putting our point of view also. But yeah, that was more on the more like a comment on their way of classification and nomenclature, that okay, it's not the only way, but obviously we are very small player in that because there is a whole dedicated team behind them. And if they are deciding it, usually they don't take it back, right? So that happened like that. But yeah, coming to your point, how we can make more of a better like nomenclature for enzymes and nomenclature for different type of genes or enzyme activities. I think people have been working towards it. The main problem right now, what is happening is that most of the annotations are mapped. Most of the annotations are mapped in the sense that, okay, if I'm sequencing an organism, E. coli, any organism, what we will do, the annotation tool will usually map it to the existing annotation, and whoever they find the best kind of hit, they will map the same annotation to this. So if the previous annotation or the best hit of one gene says that, okay, this is a histidine kinase, then that particular best hit or in the genome will also get the word histidine kinase. So I think we need a better mechanism, I think, in terms of mapping this, that the mapping should be with the either domain based mapping or some verified name mapping, because what you will see, a lot of uncharacterized proteins are increasing in all the genomes, right? So what is happening, that because it is previously uncharacterized and the best it is uncharacterized, so the mapping becomes uncharacterized. So it will say uncharacterized kinase protein, because the earlier version was saying uncharacterized kinase protein, it will also say uncharacterized kinase protein. But if you see the 10th hit, it might say some histidine kinase or like histidine kinase 4p kind of protein, or transcription regulator 4r kind of protein. So if we have a better annotation system in terms of this blast based mapping and domain HMMER or HMM scan based domain mapping, I think in that way the classification and the nomenclature of newer enzymes or potential similar enzymes will become a better way.
Perfect. Yeah. Yeah. Very well said, Gaurav. Yeah. Wonderful. So moving slightly to a further thing, Gaurav. So you have developed a wonderful SG suite. So,
Yeah, yeah.
so would you like to tell us something about
Yes,
that?
yes,
So that you
yes.
want to share your screen and please.
Yes, yes. So just to give you introduction to all the people who are here, that means I'm sure all of you are aware of Google Scholar, how it works and everything. So right now, and I'm sure many of you are prominent people in the field, and many of you have from a lot of citations, a lot of H index and all those things. So let's say that we are just a simple data analytics team, and we are just trying to bring transparency and normalization into the picture. Okay, so that's what we thought, that okay, why not think of the Google Scholar as a platform where we can do some more data analytics. So just to tell you that Google Scholar has been like this from last 20 years, frankly. Okay. And it has been exactly like this. There has been no change. So what we thought that, okay, means a lot of people boast about H-index citations, and which is great, means obviously they are doing great science. And let me just start it with the disclaimer that I'm not, bashing on anyone on their science. Okay, I'm just simply doing data analysis here. Okay, so what we have done, we have made a tool which works on your Google Scholar page on your laptop, and it will give you a very highly detailed information about your profile on the same page of Google Scholar. So you don't have to download it as a separate tool. It will directly come up on your Google Scholar profile web page, and you can see all these things. I will try to show you it quickly also. So you can download it. It's a browser extension designed to scrape key publication statistics from Google Scholar profile, providing insight into author contribution in academic work. So our main point is that right now, H-index is vague. Citations are vague. Even if you are first author in any study, or if you are 10th author, or it's a 100 author paper, everyone gets same H-index, everyone gets same citations. But that's not transparent, because first author has worked more, maybe the corresponding author has worked more, second author has worked a little bit less than first, and similarly, like 10th author or 20th author might have contributed only few hours on that particular paper or something. So we thought that, okay, can we classify this based on authorship? So what we have done means, obviously this tool does a lot of things, and I will definitely say that please try to download it and try to see on your profile, your colleague's profile. It's a very good comparative tool which exists. It does authorship based categorization based on first, second, co-author and corresponding author. Remember that Google Scholar profile is editable. You can put it there that you are the first author you are the co-first author there, you are the co-corresponding author there, based on whatever we have suggested. Okay, so like corresponding authors are star marked, co-authors are cap marked, and that is kind of standard information. And then it will categorize authorship-based citations, like how many first author papers have how many citations. Then it will also tell overall contribution based on the revised citations, because if you are a first author, you will get, if you are corresponding author, you will get 100% of those citations. But if you are first author, you will get 90% of those citations. So based on that citation matrix, it will calculate a new index, a new H-index, which we are calling at SH index. SH index is more of a scholar H index, or you can call it Sharma index, frankly. So, and then we have retraction mapping. So right now you have to go to retraction database to know that if any author has retracted papers. But on using this tool, you can directly, we have mapped the data, or our tool will map the data to the retraction database and show on the same page that, okay, this author has these many retractions. We also mentioned that who is a publication machine. And if someone is publishing more than 36 papers yearly, then we are calling it a publication machine. And obviously, we are making a lot of interactive visualizations. Just to quickly tell you, so means I'm just showing you based on my profile. We also categorize them based our papers on Scopus Journal ranking. So like how many are Q1, Q2, Q3, Q4, how many corresponding they have, how many citations all the first author, co-author, second author are getting, what is the distribution of those citations. And we also provide like more of a authorship role based contribution. So let's say if I have 21% my first author papers and I'm getting 50% citations from my first author papers, that's a good number to some extent. But you will see many people who will have a lot of co-author papers, and their citations will be coming from co-authorship. I will come back to this. You can do calculations per year, you can do cumulative, you can do in a range. Means right now, Google Scholars or Google Scholars simply show that, either you sort based on year, based on citations, but using this, you can do a lot of things. I will show you quickly. If you see what will happen, if you have good number of first author and corresponding author paper, your H index and SH index will differ just by a few number. So 13 to 12. But if you have, let's say this particular author has like almost 70 60% of papers as co-author and 90% of papers as citations. 90% author, 90% citations are being procured by 60% co-author papers, and first author and corresponding citations are very small. So their H index will drop immensely. Like if you see here 28 to 16, but the 16 number is more normalized as compared to the 28, which was a vague number. So we are trying to portray this SH index as a method of author contribution normalization, and which will be more genuine as compared to the normal H index. We are also planning to, means maybe in few days, we will also provide adjusted citation here. So these citations are total citation, but based on author contribution, we will provide adjusted citation here. Now, just to show you quickly how few of the examples work. So it also indicates retracted paper. So if you see here, this author has 6 retracted papers, so it will be visible here. And we will also showcase this retracted information on Google Scholar page like this, red marks, as you can see. Okay, and so these are all retracted papers. If someone is publishing more than, let's say, 48 papers, so I think the current version is 48. So it will showcase that this person has 48 papers per year in these years. Okay, just to quickly show you on the internet or something here, so that you can see it. So when you will download it from the tool itself, it will directly come up here as Run Scholar Lens, and you can run it, and it will run on all the profiles and showcase the result in 1 or 2 minutes. Means you have to be on that page. So because it works like one person can use only one in their Chrome browser, in their browser. You cannot do it like multiple times, this, all 5 together. You have to be on one page. Now, when you will run it, you will find all these information. So let's say this particular person has 2 retractions. And when you will see those 2 retractions, you will be able to see them in the profile here. Let's say, where is the, so this paper is retracted, right? So it will bounce as a retracted word and people can see it. Okay. One interesting thing which I was mentioning about is per year. So when you do this, it's a very interesting feature, because what will happen when you will click on this 2025, all the papers and the downneath, I think the screen is a little bit hang now because this is a bulky profile. I should have tried it on a smaller profile. You guys can hear me till now, right?
Yeah, Gaurav. Yeah, yes.
Okay, great. Yeah, so it will take like few seconds to come back to this, or maybe let me just so what will happen here if you just a minute, I think it is extracting. Let me not waste time on this because this will take some time. So basically, if you select for 2025, the Google Scholar page will automatically become for 2025. Okay, which is very interesting because there is a lot of work in that, but you will see it quickly. Just a minute. I think this paper is in this. Okay, that's good. So yeah, I will come back to this. Remember that, like many people are publishing very well. Even if they are publishing more, it doesn't mean that they are doing bad science. More number of papers is not about bad science. It's just an indicator that, okay, these people are publishing more. Like this is a very prominent researcher in San Diego, Alessandro Sate, and he has like a lot of publications in every year. He has a very big group, but he publishes very well, and in prominent paper journals like Cell, Science, and Journal of Immunology, so many wonderful work. So as I said, I'm not saying that this is bad science based on publication machine, but this gives you an idea as a person who is visiting these kind of websites. Okay, just to give you one more idea here about this particular person. So let's say he has his H index dropped from 155 to 74. Why? Because he had so many co-author papers. So he has 66% co-author papers, which is claiming 78% of the co-author citations, and his maybe corresponding author citations are very low. So that's why the SH index will drop immensely from 155 to 74. So these are like some of the examples which you guys can see using this thing. I think it's very useful for people like us who want to see what other people are doing and how they are doing it, and what kind of work they are doing, how many papers they are published. Because for a new PhD student, these are also some of the indicators that everyone wants to see. So let's say if I click on 2025, what will happen? This profile will automatically get filtered for 2025 papers. So you don't have to click on year and cite it by this. You can directly see it here. You can also see it cumulative or in a range. So if you click here that, okay, how many papers I published in 2012 to 2016. So I published only 1 or 2 papers, 3 papers, 4 papers. So it will automatically filter all this based on these criteria. So very interesting tool. I'm sure the community will like it.
Wonderful.
We have done our job ideally. So let's see how the community will respond to it. Obviously, it's a hard idea to sell because we are actually lowering the credit for everyone. Okay, so even if you have 500 citations, your adjusted citations can become like 200. So in that way, it's a disadvantage for everyone. But in a way, it has to be like this because in that way, you are getting more credit for your corresponding and first author paper as compared to just working on some small idea. And I'm sure this will be a very challenging idea to sell.
I know.
And I'm already seeing.
Not only challenging, Gaurav. I mean, on a lighter note, SH index should also be acronymed for setting up the human ego index. SH.
Yeah, that is also true.
Because many people boast that I got itna citations hai, itna H index hai,
Yeah.
itna I10 hai, itna I20 hai. So this is an accurate way, but amazing, amazing thing. I have one final question which you usually ask everyone, but there are a couple of questions from Vipin and Santosh, and of course maybe Hriday also has. Santosh, would you please unmute your microphone and quickly ask? Yeah, please.
Sir, Gaurav sir, thank you.
Yeah.
It's been wonderful to hear you. Last time I heard you at Inbix. No, I just wanted to know just like what is the exact difference between when you say pan genome, core genome, and singleton, and biologically what is their significance?
Yeah. So
Sorry to interrupt you, Gaurav and everyone. Please be as short as possible. I'm sorry for this.
Sure.
Brevity.
Yeah.
Sorry.
No worries. So basically what happens when we are talking about a community of organisms which are different at species level, strain level, or genus level, we try to combine everything together. That, okay, one organism can have 20 proteins, another 35, and another 40, but cumulatively there will be several proteins which are unique for one organism. We call them singleton. Some of them will be, present all across the organisms. We will call them core, and some of them will be present in like combination. That, okay, one organism and second organism have it, but third will not have it. So how the evolution has happened that every organism get their own way of finding a particular protein. Okay, we will utilize this particular protein for my purpose only. So in that way, that protein becomes singleton kind of protein. That, okay, only that organism is having that. So there is some evolutionary advantage for that organism by having that particular protein. Now, if something is core, it means it's a core function. Every organism need to have it. So in that way, it becomes core. Pangenome means we combine core, singleton, accessory all together and say that, okay, this is the total repertoire of the proteins across all these organisms. So in that way, it's more of a cumulation of all the proteins in single copy kind of situation.
Yeah, wonderful, Gaurav. Thank you so much, Santu, for asking that very good question. Yeah, Vipin, please unmute your microphone if you want to ask that. Yeah, please. Yeah. Vipin? Yeah. Maybe, yeah.
So hi Gaurav, yeah, I'm around.
Hi.
So just asking this, I mean, it is a general question. Like nowadays we see a lot of rankings and everything. There's a lot of competition in universities, and ultimately it is based on what is the publication and everything. So what are your thoughts? And recently Savita had some report in Science of a lot of cross-referencing. I mean, the same article is referred by 50 different people from the same institute.
Yeah,
and that is where, I mean, so that is where the idea is. And also just to everybody to know, like H-index is just one metric. And we know that we had a Nobel Prize at H-index of 29 as well. Last, I think, 2021 physics. So that is another important thing to keep in mind. But my idea is like, because of this publication metric and rankings, there is a lot of unhealthy competition that is coming in, and there's a lot of malpractices that are coming in.
Yes.
So what are your thoughts on that? Just
Yeah. So means our tool is also more of in the same direction that means how this whole idea originated was simply by this because we keep hearing people boasting about their index and citations. Okay, my idea was that can we normalize it? Means I'm sure those people are working very hard and that's why they have those many citations and all. And but the idea was that can we normalize it for everyone? Like there are these fields, let's say biological science, chemistry, which are highly prone of these kind of ideas. But other fields are in that way. There are many fields which doesn't take care of authorship. So we don't need to think of those fields because they are already not taking care of this authorship. But the point is, all these rankings and all these citations, they are more of becoming more of a tool to gain visibility. Okay, this is more of an effort to say that it is not the right strategy. And as I say, I think I don't know if it was visible on the side that metrics are stupid. What we are just trying to do, we are trying to make them less stupid. Okay, means they are actually stupid. So I'm not a fan of metrics, but means we are just trying to do some more analysis to say that there are better ways, and in those way it can make more sense as compared to whatever is existing, and it has been long existing now.
Wonderful.
Right. Absolutely. I agree.
Absolutely. Yeah. Very well said, Gaurav. Gaurav, one final question from my side. I usually ask, my guests, are there any instances where you disagreed in an agreeable way with any of your colleagues?
Disagree in terms of?
Any kind of a disagreements that you made it in a very smiling or in a very agreeable way, even with your bosses, with your colleagues. This usually I ask everyone so that young audience can learn, how to disagree in an agreeable way, because both.
Disagreements are part of every culture, frankly, and we should take disagreement in a very positive manner. Means I usually say to my lab students, especially that if you disagree with something, please come forward and talk to me. And even because it's like I'm also a human being. I can also do like some minor mistakes or something, right? So we usually try to take it in a very positive manner. I'm sure everyone takes it in a positive manner. We have to keep this ego word aside, and then there is a concept of inclusive ego. Like we need to have like more of an inclusive ego. Like don't just become me and myself kind of exclusive ego, but more of inclusive, that my ego should become this big that this whole world is ours and all. So in that way I think we should means I usually take it in a very good manner. Since we had a lot of discussion with all the colleagues, and several colleagues we also discussed about this Google Scholar Lens and these kind of techniques, because these are very in a way these kind of things create issues, because people will feel that this is targeting me. But that's why I said in the disclaimer that I'm not targeting anyone. I'm just simply doing some data analysis, and we are just trying to bring things out. Another few things which we are also working in the same direction. I'm sure we will keep you posted. And I'm trying to make like a more of a this metrics and the culture, the lab culture, how we can, how as a data scientist I can contribute in making it better as a researcher. So let's see.
Thank you so much, Gaurav. One final and a very on a very smiling question. When is your next marathon and where?
So I'm planning to do Tata Mumbai most probably in January. So excellent. Let's see.
There is also Jaipur marathon, and maybe Hyderabad marathon, is coming up in November. I'll try to participate with you. Let's see. Thank you so much, Gaurav. Today you made our day. All of you, please switch on your camera with a wonderful for a wonderful photograph with Gaurav, our guest. He has been an impeccable researcher. In summary, I've been a great fan of his wonderful and unique ways of setting up the tone for publications, especially taking up the microbial genomics to a completely different ladder. Thank you so much, Gaurav. Let's have a good photograph. All of you say cheese. हैदराबाद तक दिखाना चाहिए आपका जो चेहरा एंड आपका स्माइल. Thank you so much, and let's give a big round of applause to Gaurav, please.
Very
Thank you,
interesting.
Gaurav.
Thank you.
Yeah, thank you, Gaurav, and I'll put up this in the YouTube page, the recording. Thank you so much. Thank
And
you.
please stay in touch in case you have some concern or something. Yeah.
Yeah, thank you. Thank you, Gopesh, amid your driving, and thank you, Ansari sir. Thank you, Kausar. Thank you, everyone. Thank you, Santu and Vipin. Bye. Thank you.
Thank you, sir, for the
Okay,
conversation.
good evening.
Bye.